invitation here, and I'd just like to start out by saying that I'm going to talk mostly about new work in songbirds, but I'm going to start out by covering some published work in mouse very quickly. But all of this is tied together with a focus on interactions between motor and auditory systems. And in the mouse work, particularly secondary motor cortical interactions with primary auditory cortex, and in the bird work, vocal premotor regions that are important to song patterning and their interactions with higher level auditory areas and the role of those projections for learning. So just maybe to set the stage, especially with speech mechanisms, it's appreciated that there's a modular organization. The clinic has shown time and again that productive versus receptive aspects of speech are localized to different parts of the cortical mantle, and particularly in the left hemisphere in most individuals. But underlying those modules is a bidirectional network of fibers, including the arcuate fasciculus, which conveys information both from the auditory system to the frontal regions of the brain, and also, we think, from motor regions of the brain to the auditory cortex. And so the things, is there a sizing option I have here? Put the little last bit in there, and I can just read it. But I'll talk about motor cortical modulation of auditory cortical activity, and then motor to auditory pathways for vocal learning. And then, depending on those lights, I will talk about vocal timing mechanisms. Great, thank you. Okay, so there are many examples of how movement influences auditory processing, but probably the most compelling evidence is from recordings made in vocalizing marmosets from the primary auditory cortex of these animals by Stephen Eliotis and Xiaoxing Wang that showed a vocalization-related suppression of auditory cortical activity. And in fact, the timing of this precedes the onset of vocalization, suggestive of a motor origin. And this and other observations, including similar effects in human speakers, has led to the idea that there's a motor to auditory cortical interaction. But in fact, the cellular and circuit mechanisms of that interaction have remained fairly obscure. So in the first part of the talk, what I want to cover is looking at the circuitry that underlies this movement-related modulation of auditory cortex in a mouse model, and address these questions, including the membrane potential signature of movement in the auditory cortex, the cellular and synaptic effectors of this movement-related modulation, and the sources of these signals, or at least some of the sources. So using either head-fixed mice, as shown on the left, awake moving mice on the left, or tethered but otherwise unrestrained mice on the right, we made intracellular recordings from auditory cortical neurons during rest and movement. The take-home is that membrane potential dynamics here occur in clamp record from an excitatory cell in the auditory cortex. Change in pattern undergoes slight depolarization and a decrease in variance that precedes movement onset and persists for some time after movement offset. And when we started these experiments, we were focused in recording and vocalizing animals, but we actually quickly recognized that this kind of movement-related effect can be seen generically. That is, any evidence of movement that we could find, whether it be locomotor or grooming, sniffing, paw movements, posturing, all of these were associated with changes in membrane potential dynamics. In conditions where we had a male mouse investigating and chasing after a female mouse, which was the most reliable for inducing vocalization, we could see movement-related shifts in dynamics that persisted during the vocal bout. So this seems to be a relatively general feature of the auditory cortex in the mouse, that is, a wide range of movements modulate the auditory cortex. And in fact, this picture is also emerging in the human brain. So for example, hand gestures, unilateral hand gestures modulate auditory cortical dynamics in the contralateral hemisphere, even in non-musicians, although those effects were first described in trained musicians. So with respect to the effects on auditory responsiveness, if you use probe tones during rest and movement. There's a decrement in the tone evoke responsiveness during movement that, in a sense, resembles the speaker-related suppression of auditory cortical activity that's been seen by Xiaoxing Wang's group and others. And to localize this effect, use an optogenetic strategy. Of course, the auditory neuroaxis is extensive, and there are likely many sites where movement-related signals can impinge on auditory processing. And in fact, there's an acoustic reflex that engages the tensor tympani and stapedius muscles. So there must be effects from the periphery all the way to the cortex. And the question is how much of it can be localized to the cortex, if any. So using an optogenetic strategy to selectively activate auditory thalamic terminals in the auditory cortex during rest and movement, we could estimate that around half of the movement-related suppression had a cortical origin. And by recording and injecting current into the cell while the animal is either at rest or moving, we can see a decrease in the postsynaptic impedance, the shunt. And using multi-electrode array recordings in auditory cortex and then optogenetically identifying interneurons, which are shown in green here, and excitatory cells shown in black, we could chart the membrane or the action potential dynamics relative to movement onset and see an increase in inhibitory cell activity that preceded and was coupled to the decrease in excitatory cell action potential activity. So this is all suggestive of a feed-forward or inhibitory mechanism in which interneuron activity is recruited during movement to shunt through the postsynaptic membrane the excitability of excitatory cells in the auditory cortex. With respect to sources, one major source that we identified using retrograde viral tracing methods is a large population of cells in both the superficial and deep layers of the secondary motor cortex. And antegrade tracing with viruses identified terminations in auditory cortex, including close appositions on RW positive cells. Again, turning to multi-electrode array recording now in the secondary motor cortex, we could see an increase in action potential activity that preceded any of the changes in action potential or membrane potential dynamics that we recorded in auditory cortex, consistent with the idea that M2 could be one source of drive to the local inhibitory circuit that generates these movement-related effects. And so to look at this, again, in awake but resting mice, we selectively activated these secondary motor cortical terminals. You see these rest-like dynamics, high variability. When the light turns on, there's a signature that, for all intents and purposes, looks like what we record during movement. That is a decreased variance and a slight depolarization from the mean value at rest. Similarly, if we probe during either lights off or lights on, the tone evoke responsiveness is decremented during the lights on phase, consistent with M2 being a source of movement-related activity that's driving this decrease in auditory responsiveness. Again, using an optogenetic approach to selectively turn off these secondary motor cortical neurons, which we could do very effectively, as we confirmed with multi-electrode array recordings in M2, we could then probe this effect in auditory cortex. So this is an intracellular recording in auditory cortex during a movement phase. We turn on the light. Within about 500 milliseconds, that stops the animal from moving, but there's a period while the animal continues to move, and we can see that the membrane potential dynamics have been restored to a rest-like state. And this is just a cumulative distribution of these effects, but every time we turned on the light and silenced M2, the ipsilateral auditory cortex returned to a rest-like state prior to movement offset. We could also probe this transition while the animal was still moving, but the auditory cortex was in this rest-like dynamical state. And sure enough, the tone evoke responsiveness had recovered not completely, but to a degree we would have estimated based on the relative cortical contribution to this effect. So again, this is just a quick review of published work, but there is a signature in the mouse auditory cortex of movement. It involves a slight decrease in resting membrane potential and a decrease in membrane potential variance. Both of these are accompanied by a decreased auditory responsiveness. So in that sense, it resembles what's been described for vocalization-related effects in our own auditory cortices. The players in this, at least local to the auditory cortex, is a feed-forwarded inhibitory pathway that involves in part valvular depositor cells acting postsynaptically. And a major driver of this is a population of superficial and deep layer cells in secondary motor cortex that project to auditory cortex. And I should add, although I didn't show this here, that through intersectional tracing studies, we were able to show that a subset of these M2 terminals, or M2 cells that project to the auditory cortex, also target deep brainstem structures associated with movement. So it's consistent with the idea that these cells are providing a corollary discharge signal that can inform the auditory cortex of upcoming and ongoing movements. Okay, so to turn a little bit more to the models that we discussed today, this is from a review by Hickok and colleagues of forward interactions in speech. As we've already covered several times this morning, the idea is a controller system can not only manipulate the vocal tract and generate vocal output, generate some auditory feedback signal, but through a corollary discharge mechanism, an internal model of the vocal tract can use this information to serve as a means of comparison to the actual auditory feedback signal for error detection purposes that can then be used iteratively to drive the controller system adaptively to affect learning. So one challenge with mice is that they don't learn to vocalize, and in fact their behavioral repertoire for certain kinds of auditory guided learning is somewhat impoverished. Another focus of our lab's work is the songbird, here a pair of zebra finches I'll tell you more about in a minute, but these are really the consummate vocal learners, outside of a few other kind of lab-unfriendly models like toothed whales. These are really the best for studying vocal learning in a vertebrate. And they culturally transmit their vocal patterns from one generation to the next. And I'll just play this little video clip so you can get an idea. We can spare the audience. Teenagers, this is a very useful model for parent-teen interactions. He does fall asleep, yeah. This is actually an aggressive display between the tutor and the pupil. And it's common actually for, especially in early instances of this interaction, for the pupil to fall asleep. After the adult male sings this song to him. The adult doesn't engage in any kind of motherese. This is just straight on, full bore song. And even though the juvenile fell asleep in this instant, this is really a transformative experience for him. Here's a sonogram of the tutor song. This is the inchoate song that the pupil, or vocalization that the pupil makes prior to tutoring. And if you record the adult song of that pupil, it will be an extremely good match to that tutor song, even though its exposure to the tutor can be limited to just a very, very early stage in development. And in fact, in this timeline, there's a sensitive period for memorizing the tutor song, typically which ends around 60 days after hatching in most songbird species, and then a very prolonged vocal hatching phase. And this is also species dependent. But maybe just to emphasize, this is a very, very rapid phase of total auditory experience necessary for accurate copying, maybe on the order of a few hundred seconds of tutor song. This is really laborious. The animal will undergo maybe 100,000 trials of singing. The pupil will sing over, several thousand times a day for a couple of months to generate something that is a close copy of the tutor song. And in the zebra finches and some other songbird species, the song becomes highly stable with the onset of sexual maturity, and in fact, that stability is essential for functioning as a communication signal, especially to females of the species. But that stable pattern is maintained using auditory feedback, and I'll come back to that a little bit later. The circuitry is also very well mapped out in some regards, and particularly for motor control. And I'll just simplify this. There's a premotor network that includes this structure HVC, which sits at the dorsum of the telencephalon. It projects to a motor cortical analog, here known as RA, which projects in turn to the vocal respiratory planet and brainstem. And this pathway is obligatory for producing motor vocalizations in birds. And again, by just very rough analogy, you can think about HVC as kind of like Broca's region. If it's bilaterally lesioned, then these birds can make their contact calls and innate calls perfectly well, but they can't produce these learned songs. HVC has a projection cell type, premotor cell that projects to RA, then it has another cell type that projects into the basal ganglia, and this pathway isn't shown in completion, but this projection isn't necessary for production of adult song, but it is necessary for feedback-dependent modulation of song, the implication that this pathway is playing a critical role in learning. And finally, there are ascending auditory projections through primary and secondary auditory regions of the telencephalon that project into HVC. And in fact, in different states, auditory activity can be recorded throughout this network, indicating that there is feed-forward flow of auditory information into the system. And in fact, this is a simplified diagram from a review I wrote, so I'm to blame, but the long-standing impression has been that most of the architecture of this is feed-forward in the sensory domain and top-down in the motor domain, and both of those ideas are actually wrong. And I'll just develop this a little bit here now. So if we look at these regions, these auditory regions, provide input into HVC, there's a structure sitting right here known as avalanche that projects into HVC, but also receives a small terminal projection from HVC, and in turn projects to regions that Michael Fee and others have mapped as part of an error correction circuit that feeds through midbrain dopamine neurons and into this basal ganglia structure known as area X. When we started to look at this, we thought, well, the dominant view was this was a very sparse projection, probably not important to birdsong. And because it branched off of the pathway that fed into the striatum, we thought, well, maybe these are simply collaterals of these corticostriatal neurons, these so-called HVCX neurons. We used triple labeling techniques to map the different cell types, so retrograde tracing from RA and X, from avalanche, and these quickly revealed that the cells that project into this auditory region are the same cell type. They're shown in red here and a little bit higher magnification here on the right, but there were no overlapping colors in any of these cell populations. They're all distinct, although synaptically connected in some interesting ways that I'll come back to in a minute. These cells are also very sparse. They make up about 1% of the total cell population in HVC, or maybe even fewer than that. And they tile HVC, as far as we can tell, fairly uniformly, but at a fairly great distance from one another. So much lower density than the other two projection neuron types. We're able to adapt an intersectional genetic ablation technique developed by Nirao Shah and his graduate student Cindy Yang. It uses a flex caspase that we can introduce non-specifically into HVC, but in the absence of the pre-recombinase, this is non-lethal. And then use a retrogradely transported previrus to selectively express caspase in this subset of neurons that project from HVC into the auditory system. So using that design, we were able to selectively ablate these cells at early stages of development. I should say with these AAV vectors we use, it typically takes about 20 to 30 days for viral expression to ramp up. So the injections were made between 30 and 40 days of age after hatching, and we think that full expression is probably reached about day 60, the end of this sensitive period for memorizing a tutor song. And what I mean to imply by that is that the animal's housed with a tutor throughout this time, so we think it's likely that the auditory experience of the tutor was probably not so much affected as the subsequent vocal copying phases. We can then raise these birds up to adulthood and match the similarity of their songs to their tutor, and these birds with the selective genetic ablation of these HVC avalanche cells, these motor to auditory cells, do a very, very poor job of copying this. We do a post hoc analysis of the number of surviving cells, of this subtype, these HPC-AD cells, there seems to be a steep drop-off that when you get below a certain complement of these neurons, learning is more or less derailed. And I should explain these metrics a little bit. Unrelated birds in our colony will typically score at about this level, about 35%. So this, we think, is a learning floor. And normal copying levels are around 80 to 90%. Just inspecting some of these sonograms to give you a flavor for the behavior. Here's the tutor song, here's an intact sibling, and then here are two siblings from that same clutch, male siblings that had this manipulation to selectively ablate these HPC avalanche cells. And some show some elements of copying, but others, like this one, show the kinds of songs that to an aficionado are reminiscent of birds that have been raised in the absence of a tutor altogether. So it really looks like they're not copying well at all. And this might be explained simply that we're selectively ablating a cell that's embedded in a premotor network and damaging motor control. But if we make similar lesions in adults, we don't see evidence of a motor impairment. So even at long time points post-ablation of these cells in an adult, the song remains highly stable. And in fact, if anything, it seems to be a little bit more stable than what birds will normally produce. And that made us wonder whether or not this cell type was important to ongoing maintenance of the song, and particularly feedback-dependent maintenance of the song. As I mentioned, adult zebra finches use auditory feedback to maintain a stable vocal signature, and you can see that, for example, by recording a sonogram in an adult bird, then bilaterally deafening it through cochlear extirpation, and then recording its song 8 weeks later. And there are changes both to the spectral features of the song and also to the temporal organization that are consistent with the idea that the bird needs an intact auditory periphery to maintain that stable vocal signature. Again, in ways paralleling our own continued dependence on auditory feedback for stable speech in adulthood. In animals with the selective ablation of these avalanche cells, I should say, these cells were knocked out while the animals, after the animals had reached adulthood. So these are birds that have already learned to sing effectively. Deafening doesn't have this effect. The song remains highly stable, suggesting that this projection, that is, this projection from HPC into the auditory system, is critical for auditory feedback-dependent maintenance as well as experience-dependent modification of song during juvenile stages of development. So just something about the circuitry here. We can go into a brain slice preparation and make visually identified whole cell recordings of these neurons, and then stimulate these two major outflow tracts, the one that goes to RA and carries these HPC RA axons, or the ones that go from HPC to the striatal pallidum. And we know from previous studies that both of these cell types have local axon collaterals that in fact connect these RA and X projecting cells to each other. And we can see in this sort of a stimulation experiment that we could get a very nice, robust excitatory synaptic current in these avalanche projecting cells while stimulating this RA out, this HPC to RA outflow tract. But in contrast, if we stimulated in this corticostriatal pathway, we didn't detect any responses. And this is just one example, but we've repeated this many times. And it seems that these avalanche projecting cells are receiving selective input from the premotor pool. That is the pool that projects to RA and is thought to be carrying the premotor commands for singing. I think this is very consistent with a corollary discharge mechanism in the auditory cortex of the bird. And in fact, if we and others who recorded extracellularly in this region, downstream regions of the auditory system, which are reciprocally interconnected with avalanche, and one can see evidence of such corollary discharge signal, including vocalization-related activity that occurs before vocal onset and persists following deafening. I'll just give you one example or one taste of how we're trying to explore this. And these cells represent a challenge for electrophysiology because they're so sparse. So we've used an intersectional method to express GCAMP. I'll just show you this kind of experiment. This is using this lightweight miniature one-photon microscope developed by Mark Spencer and his colleagues. And we can express GCAMP in these avalanche projecting cells and then chord during bouts of singing. These are very crude and very preliminary, but you get a sense of the activity patterns here, especially if you enter this field of birds start singing in a moment. Look at this activity either with hearing intact and then after deafening, we can see that the profile, that is the elevation activity before vocal onset, high level during vocalization, is similar. In controls where we don't express GCAMP, we don't see any evidence of an autofluorescent signal. So we think this supports the notion that these cells are providing a motor-related signal into the auditory system. And I'll leave it at that, but I think the next step, obviously, is to understand what the computation is downstream of HPC, and particularly in this region that these cells terminate in. But in terms of their role in learning, I think it's clear that they're important both to juvenile learning and auditory feedback and habit maintenance. So I have a few more minutes, I think. We're doing? Yeah. So I just want to turn to another set of experiments that ask what the controller system itself is doing. And this builds on some really beautiful work from Michael Fee and his colleagues, including earlier work he did at Bell Labs before coming to MIT. And using recordings from anadromically identified HBCRA cells, Richard Hollis or Alex Kashevnikov and Michael showed that there was this ultra-sparse pattern of activity. So maybe on practice, here's the oscillogram from 3 motifs in a row produced by a bird. A motif is a collection of syllables, the sonic fixed sequence. You can see for Finch's produced. Here's the extracellular reporting from an individual HBCRA cell. This is on an expanded timescale, so you can see these really prodigious high firing frequencies that occur during these very, very sparse bursts. And single cells fired at only one time in the motif. Different cells fire at different times in the motif, leading to this notion of a feed-forward mechanism involving a chain of interconnected HBCRA cells where activity propagates in a clock-like fashion through this network to control song timing. And this is a very, very influential and very attractive idea. And to directly test this, Michael Long, while he was a postdoc with Michael Fee, used a miniature Peltier device to bilaterally cool HBC, which produced a remarkable effect of uniformly stretching song tempo over all time scales. So simply, if you took the control song and stretched it in Photoshop, it would exactly match both the finest time scales and the longest time scales, the actual cooled version that was produced when the Peltier was turned on full power. And we approached this, we thought, well, this is really amazing, and I've had many conversations with Michael and his group about this, and what I was curious about is the synaptic architecture that can support that kind of process. And we being Kosuke Hamaguchi, a former postdoc in my group, I used a miniature Peltier device similar to the one that was employed by Michael Fee's group, and reproduced this observation. That is, when HPC is bilaterally cooled, the song stretches, and it stretches about 30% for every 10 degrees Celsius cooling, which yields a Q10 of about 1.3. We thought, well, we can go in and measure the network in HPC and look at its temperature dependence. And we thought, this is either going to be really, really exciting because all the other studies, and there are about 150 of them, that have studied temperature dependence of synaptic transmission in networks reported Q10 that's about an order of magnitude higher. So we thought maybe there are going to be very special temperature-dependent synapses in HPC, or maybe there's something else going on here. So the design of these experiments involve recording synesthetized birds. An intracellular recording is made from an HBCRA cell, and then we anadromically activated the outflow tract that connects HBC to RA. And this reliably evokes the synaptic potential. We can then cool HBC and study what happens to the onset delay. And it returns a value that's about 5 to 6 times greater than the behavioral effect. So the HBCR to HBCR synapses show a temperature dependence that dilates about 150% per 10 degrees Celsius as opposed to 25 to 30% for the behavior. The question is, what's the architecture that can explain that? And so one thing that's not emphasized perhaps enough is that the recurrent architecture in the vertebrate brain is also present in the songbird. In the case of the song system, there's a projection down to the vocal respiratory gland, as I described. But from a respiratory premotor complex in the lateral medulla, there's a bilateral projection up through the thalamus that feeds back both ipsilaterally and contralaterally to HVC. And in fact, this is a critical linkage for helping coordinate activity in the two hemispheres, and particularly coordinating activity in the two HVCs. So if you simply cool the system bilaterally at HVC or unilaterally on the side that you're making the intracellular reporting and stimulate it on the contralateral side, you can measure the propagation time through this network to a minimum of 4 synapse loop. And that temperature dependence exactly maps on to the behavioral temperature dependence. So the raw times for activity propagating through this network are about 16 milliseconds to 38 degrees Celsius. And with cooling, stretch about 4 milliseconds to about 20 milliseconds, which yields a Q time of about 1.25, which is very, very close to the behavioral effects just shown here. So again, the HVC-RA synaptic network, which has been implicated as a backbone for a SynFire chain, has a temperature dependence that's much greater than the behavioral effects. But that temperature dependence and behavior very closely maps onto the network level, that is the brain-wide temperature dependence, suggesting rather than a local model that if there is a SynFire chain, it's a loop-like structure that passes out of the forebrain into the brainstem and back up through HVC. And we can estimate that HVC might contribute 1 or 2 additional synapses to the delay. So I'll finish there just to emphasize what I've told you about synaptic and circuit mechanisms for corollary discharge in the auditory cortex, and an important role for secondary motor cortical neurons in driving feed-forward inhibition in the auditory cortex that underlies movement-related modulation of auditory cortical activity. An analogous pathway in birds that conveys information from a premotor region towards singing and song patterning into the auditory system that plays a learning-specific role. And in both cases, we're very interested in knowing what the downstream consequences of these projections are for modulating auditory feedback signals or interacting with auditory feedback signals to facilitate error detection. Okay. And just to emphasize the include of the work, Todd Robertson now has his own lab at UT Southwestern. And a student in my lab, Aaron Heisey, has been leading these avalanche studies. And then David Snyder and Anders Nelson have done this work on mouse auditory cortical circuits. And Kosuke Hamaguchi, who's now at Kyoto University, the prime hand of the timing experiments that I talked about. Thanks.